Greetings. I'm Yvonne Stapp for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Most of us don't think about ordinary physical movement until something goes wrong, and the complexity of movement is our topic today. Our guest is Jill Crittenden, scientific advisor and research scientist at the McGovern Institute for Brain Research at MIT. Dr. Crittenden explains how the brain organizes normal movement and how disruptions in the brain circuits result in disorders such as Parkinson's and Huntington's diseases and other kinds of motor dysfunctions. She also describes some of the advanced technology and genetic engineering that is enabling this very advanced research. We are very pleased to welcome Dr. Jill Crittenden, and thank you so much for being with us, Dr. Crittenden.
My pleasure, Yvonne. Thank you for having me.
And I would like to start with a little background because, like I say, you're aware, far more aware, that we don't normally think about how complex ordinary movement is. But it is a very complex thing. Could you give us a little bit of background of how the brain does this for us?
Yeah, it's quite fascinating, right? Because there are many things that we're just born to do, or that just happen automatically, that are motor movements like heartbeat, blinking, or even a baby walking or suckling.
Yeah.
Those are very, very natural things. But there are other things that it takes us years to learn how to do carefully. And one of my favorite examples is eating with a toddler, right? Who's trying to
Yes.
even just with their hands, let alone chopsticks or a knife and fork.
True, great example.
And years, it takes years to do that. And I think another interesting sociological aspect of that is that if we don't do it right, the social feedback is huge, right? If we have something stuck in our teeth, there's nothing worse than that, than having something on your mouth, right? That you didn't get it in your mouth just perfectly. So I think it takes years to get those things that we don't, once we've learned how to do it, we think it's simple, just like you said in the introduction, but in fact takes years. And there's a lot of punishment and reward that go along with doing it right or wrong.
Right. Now, the brain has like specific areas, I guess, for controlling this, but it sounds from your research that it is actually very complicated. It is the neurochemical basis behind that. A lot of things can go wrong, but can you give us, do you have any images to show us where this is organized in the brain? And
Yeah, let me share my screen. So this shows the human brain, right? So your eyes would be right here in front. This is the famous neocortex, right? That's involved in moral planning and thinking about the future far, far down the line. And under that is actually a really big area that is what I've spent many years studying with Anne Grabiel at MIT. And this area is called the basal ganglia. And it receives input from all areas of the cerebral cortex that's overlying it. And I think one way to think about it is that when we learn many motor action plans, right, we can do so many different things. We can ride a bicycle, play the piano, beat ourselves like we were talking about. There's just thousands of these motor programs that it took a long time to figure out which muscles to move in what order and how. And once we have those action plans in our brain, the idea is that these plans are stored in the neocortex, that this underlying area helps us learn them, and then they get relegated as these sort of chunked sequences of movement that are stored in the neocortex. And one way I like to think about the other job of this basal ganglia, besides helping us learn how to do things, we know it's not only important for learning, but it's important for choosing which motor plans to execute when. And so that's a complicated phenomenon. And one way I like to think about it is that there's just all these action plans here in the neocortex, and they all want their action plan, right, to be to be engaged. So they're always have these connections to the basal ganglia, but the basal ganglia does what people in the field call action selection. So it integrates things like mood, whether you're hungry, whether you're stressed, the environment that you're in, whether there's pizza available. You know, you're not going to engage the eating action plan if there's no food available. So it also has a lot of information about the current context that you're in, as well as memory, with whether the last time you engaged that motor action plan, how did it work out for you? Did you get sick? Did your mom tell you not to do that? That kind of thing. And so we think that that's a big part of this underlying area, is to figure out which of all those different motor action plans that are connected to it should be engaged in the current situation. So it's like a battle to control our behavior.
Yeah, that's very interesting that you refer to this as plans. And so it's like a little menus or recipes for how to do this, how to do that, the stuff that we don't think about, and that you notice it in normal circumstances, maybe like when you see that small children age 5 are learning to manipulate a pencil or whatever. And that is very difficult until there's a certain level of maturity and so on. And we've all been through this learning instruments or riding a bike, as you mentioned, that sort of thing. But there's a lot involved in this. Can you tell us the kinds of that there's neurochemistry evidently behind this as well? Then all this leads up to things that can go wrong. Could you give us an idea of how that works, how the neurochemistry is involved, and then maybe how things start to break down?
Yeah, the neurochemistry is, as you might guess, extremely complicated. There are many neurochemicals and many neurotransmitters in this region. And one neurochemical, GABA, actually inhibits neurons from firing. So we call it firing because they actually have a voltage potential that travels down this long thing called an axon. Actually, these images behind me it's a sculpture in the building in which I work, in the McGovern. And those long connections, each of these is a neuron. And so a neuron has a cell body where the DNA is and kind of, you know, what keeps the cell alive. But it has these very long connections, all of them, many of them. Some of them make as many as 100,000 input connections. It's like the universe, right? Trying to think about how many complex connections there are. And so they send signals, electrical signals, down those long connections to another neuron that they want to impact. And one of those signals can actually be inhibitory. So it can turn off the other cells that it's connected to. And I think that's a fascinating thing also about this basal ganglia, is that it's very enriched in GABA, which is an inhibitory neuron. And of course, maintaining a voltage potential takes energy, because that's what it is, right? It's holding an abnormal ion flow across the membrane. And so there's actually a huge amount of sugar energy burned in this part of the brain to keep it off. And so that GABA signal is really important, as you can imagine, because there's all these motor action plans coming in as potentially executable. And in order to have any meaningful way in the world, you need to keep most of those off, right? You want to have one particular motor action plan, not a bunch of random ones that are competing with what you want to do.
That's very, very interesting. And this is one reason why they call the brain, the human brain, the most complicated thing that we know of in the universe, I guess. But you describe so well the complications of this, the choices, the minute choices that the chemistry and the circuits are making all the time, and so on. All right, what can go wrong? Then you investigate diseases, you investigate other kinds of disorders that are due to, say, drugs, to other kinds of things, maybe injuries. Can you give us an idea of what can go wrong and maybe how that is processed in the brain?
Yeah, so a lot can go wrong. And this area, for some reason, is often implicated in brain disorders. And some of the most widely used drugs for brain disorders impact the dopamine system. And dopamine is highly enriched in this structure. It's what's missing in Parkinson's disease. So actually dopamine is produced
Ah,
far down here in a small area, and then it's broadcast widely across the brain, but especially to this area, to this motor control area. And so when you don't have dopamine, then this motor control area can't correctly motivate movements. So people with Parkinson's disease often want to stand up, for example, and it's difficult to get that going because the sort of this go signal from the dopamine isn't driving the
ah,
basal ganglia properly. And actually looking at people who had these symptoms and then at their brains after they had died long ago gave a huge clues to what was going on in this part of the brain. And interestingly, other people have a disorder called Huntington's disease where they have too much movement. So they have something called Huntington's chorea, which is a word for dance. So they have a lot of just abnormal dance-like movements. And when people compared the brains of people who had died with Huntington's disease and those who had died with Parkinson's disease, they came up with a really interesting model of the basal ganglia that in the past 20 years has actually been testable and shown to be basically right. People argue about all the details, but it's
yeah.
amazing that it's actually basically right, because with genetically engineered mice, we can really test it, right? It's not just looking and seeing, okay, this person had a hard time moving and here's what looked like was wrong in their brain. You can actually make those changes in a living mouse and say, well, what happens when we drive this pathway with dopamine? Does the animal move more? And they do.
Mhm.
And so based on that, this model of the basal ganglia having two main pathways, one for doing something and one for not doing something.
Mm.
And so that kind of feeds back to what I was originally saying. A lot of what the basal ganglia is doing is saying, don't do this thing right now. And it has to actively inhibit things that are around the area that is responsible for engaging the selected motor action. So dopamine is important for getting things going properly. And so one major therapy still for people with Parkinson's is dopamine replacement therapy. So carbidopa is is taken orally, and people can have too much movement actually, because now you're replacing the missing neurochemical, but it's not precisely controlled the way it should be because you're just taking a pill. And so then sometimes you get things that are called dyskinesias, which Michael J. Fox has talked about and kindly shown videos of that problem. And in some ways, those resemble Huntington's disease, these just abnormal limb movements that appear useless and uncontrollable.
This is very interesting that these diseases have these direct, like chemical, what do I want to say, chemical dysfunctions or loss or too much or whatever. And that's disease. And there are other kinds of things. I think it's rather interesting that you have worked on drugs and food issues that also cause problems with the motor systems. Could you tell us something about this?
Yeah, so actually every drug of abuse activates the dopamine system or somehow potentiates it. So it can either directly activate it or it can prevent it from being turned off. These different drugs, amphetamine, cocaine, morphine, nicotine, marijuana, all of them, alcohol, all of them work on this system. And there's two aspects to that are really interesting. One, people often have abnormal movements at some point when they're using drugs of abuse. So for example, a lot of people use amphetamine like fighter pilots and stuff, right? To have extra energy, to have more
Oh,
drive of the motor system. But at the same time, it's engaging actually this system of the brain that really knows how to do a motor action over and over again, very precisely, like we were talking about. So another way to think about these complex motor sequences, people call them motor habits, because you don't
yeah.
even have to think about it, right? You can,
Routine, yeah.
yeah, you can drive your car without thinking about where you have to do this, such and so. And that means you can free up other parts of your brain to think about, what you're going to buy at the store when you get there or something. You don't have to think about how to get to the store anymore. That's already on autopilot. But another way to think about it is that autopilot might just take over sometimes, right? It might be activated. It's nice to not have to control it all the time so that you can use your neocortex for other things, but you want to be able to control it in other situations. And that's kind of what people think is going wrong with these repetitive movement disorders. So for example, in autism, people will often have these repetitive movements that
Oh, yes.
people become so engaged in them that it's hard to disrupt them. And the same thing with drug addiction. People will become so engaged in this repetitive motor habit, in part because they've just driven this dopamine habit motor system abnormally with this drug.
Can you please give an example of these repetitive movements that if people have not seen them in autism or in drug disorders, I think of like with alcohol, people stagger, they can't control their movements so well. But I think we're less familiar with drug and with autism, those what you mentioned, repetitive kinds of motions. What are those?
Yeah, so there are quite a variety of such movements. A common one for kids with autism, they call stimming, when kids flap their hands, for example, for a long period.
Oh,
But they can also be very complicated. For example, people might like to sort buttons or something like that, right? And they just get really involved in these tiny tasks and are hard to disrupt, and they seem useless. I think that's one way of defining them, is that there doesn't seem to be any reason that they get engaged in these things. For example, OCD, part of the definition is that I haven't checked the DSM recently, but it used to be if somebody does it for 45 minutes or longer, then that can be categorized as OCD. And I like that example because it shows what a spectrum we're all on, right? Like, okay, I do this useless thing for 30 minutes and it doesn't matter. Or we do a lot of things like nail biting, right? That's kind of useless. So I think we're all on a spectrum of these,
yes.
having these repetitive, seemingly useless movements. And one thing that was really fascinating talking to pediatricians is that it's a normal part of toddler development to have these repetitive movements, like hand flapping. And so one thought is that just in autism, that the people don't grow out of it. They just keep engaging those things. But even though it was a normal part of the pattern of motor learning, or for
Or
whatever
other
reason.
movement becomes entirely normal, but then you have these weird kinds of things there, distortions like that. That's quite interesting. And the same, I take it, with the drug, alcohol, all of those kinds of things can cause this, right? A similar problem. That's
Yeah, of varying types.
okay.
So for example, people that are using amphetamine or cocaine, I think also like to do very fine motor movements. So like sometimes skin picking is something that
Oh,
those people
wow.
will do. And those people can get those same motor actions with high stress situations. So for example,
Yeah.
solitary confinement in prisons, animals in zoos will do these things. So just the neurochemicals become imbalanced, and it becomes difficult to control these repetitive behaviors.
That is really fascinating, that with these things that we never really think about on a day-to-day basis. And may I ask one more question before we leave that and go to the research part of it? Does this have to do with other things that are procedural learning as well? Step 1, step 2, step 3. I think of computer applications, processes. You know, mentally you go step 1, step 2, step 3. Is that related to this kind of motor thing, or is that totally separated? In other words, procedural learning is quite different from some other types of learning, but so I don't know if it is related to this mechanical thing.
Right. So there are different ways of chunking behaviors, right? So some things I think we do start with a procedure like how to ride a bike, and it
Yeah.
does really need to be in this particular
Yes.
sequence that you do it. But there are other things like making soup, right? And it also involves a complex sequence of movements, but it doesn't have to be so very precise that it becomes a habit movement.
Right. Yeah.
So I think there are different types of
Oh,
movement sequences that we have, and some become this kind of non-procedural where they're just automatic
yes.
because they have to be so precisely executed in a very particular pattern that we do over and over again. And some things just never reach that kind of automaton because we don't have to do it in that very precise pattern and timing and so forth. So it never engages the same motor habit system.
Yes, I see. Then that's good to know, to get that cleared up. And now can we go to your research? You mentioned those mice that volunteer their time and effort and their little brains. How do you proceed to look at this kind of stuff, because it's so complicated? Can you give us an idea of how this research develops or works?
Yeah, so there's quite a big group at MIT studying this. And I think I'd like to introduce a little video just actually of my own
Oh,
kid
sure.
that shows the way in which we study how motor sequences that are rewarded become imprinted in the brain. And so I think one way in which you can reward mice is they really like chocolate. And so you can teach them a maze. You can teach them in a maze, and you can teach them, if you hear a high pitch sound, then the chocolate's going to be on the right when you get down to the turning part of the maze. If you're a low pitch sound, then the chocolate's going to be on the left. And then initially, right, they're just trying to like look around the maze. They don't know there's any chocolate, and they do all these non-habitual motor behaviors, just exploration. But eventually they learn the task and they really exploit it and get, they do. One of my colleagues made a videotape of a rat walking down the maze, and it was putting its foot in exactly the same spot every time after this had basically become a motor habit, so that it just automatically executes, right, to do this particular procedure. And I wanted to share an example of how learning a motor task can be rewarding. And this is engaging the dopamine system, right? The dopamine system is saying, release dopamine so you can make that habit. What you just did is rewarding, so reinforce that particular circuit. And that's the kind of thing we're doing in mice, is recording from the neurons that are activated by dopamine and other neurochemicals. I mentioned GABA, glutamate, acetylcholine, serotonin, endogenous opiates. I mean, it's a real potpourri in there that are controlling all these neurons. But we're trying to figure out which circuit, which neurochemicals, which cells are releasing them, that sort of thing in the brains of mice that we can genetically engineer. So it's not just an observational science. We can determine cause and effect. But here's an observation, right, that we can make to see that my daughter's learning how to
Oh, yeah.
So she does the exact same thing,
Joy.
puts her hand up there, you know, she's like, I need to do exactly the same thing I just
Yes,
did because
yes.
the lid opened and that was so
Right, right,
rewarding.
right. Isn't that interesting, that association?
And so all these brain regions, right, are encoded.
Yes,
And it's just a matter of combining them in the right way to have these
yes.
little chunks of action plans that are encoded in the brain.
And it takes time to develop those skills. And I think a lot of people who remember maybe not learning how to walk, but learning to play an instrument or learning the movements in a sport like golf or tennis or any number of things, that it's very complicated and it takes time. And so could you explain, by the way, since you've got the basal ganglia up there, these weird images here of hand and foot at the top?
Yeah, so people's a long time ago recorded from different areas of the brain in animals
Yeah.
and even humans, and found out that, you know, if you stimulate somebody's lip, for example, right, then you can see activation here and vice versa. These areas can control it. And so there are these homunculi that are actually outlined here. And so the fact that it's organized somatotopically to some degree is a perfect example of how complicated these circuits and how overlapping these motor sequence circuits are.
Yeah.
Because anything we do involves, you know, using, you know, opening your mouth at the same time as your fingers are coming towards it if you're going to bite pizza or something. So you need to have these. It's hard to figure out the circuit because it's got part over here, and
Yes.
depends on how hard you're going to have to bite. And so,
yeah.
okay.
Like then to go into the research a little more, also that you, the way that you proceed to do this, you use mice. And I know that many people think, well, mice are a little different from humans. So can you say that in terms of the mouse thing, in terms of the mouse brain and human brain, that there are these equivalents as you sort of described? How do you use this research? And I then want to get in some other areas of how you go about it too. So is there an equivalence good enough between mice? Reassure us.
Yeah, I think it depends on what you're studying. So
Mhm.
one thing that makes humans really special is that we can plan for things 20 years in advance, right? We do things where we're not going to have a reward for 20 years, go to school.
Yeah, right.
You know, court someone we love because we think it's going to be a long-term relationship, and mice can't do that. They don't have such big brains to really predict the future many, many years in advance. So I think that they're not useful for that, for understanding those kinds of challenges that we face, for modeling those kinds of disorders where people aren't good at planning for the future, for example. But they're excellent for studying basic motor learning. So if you look at baby mice, I have videotapes of baby mice learning how to walk, and you can see all the kind of typical things that you see with baby humans.
Yes.
So I think they're great for learning how the motor system develops and is controlled.
Right.
Because that's conserved.
And you use the mice as a model, and you use genetic engineering and other forms, sophisticated kinds of modeling. Can you explain that just a little bit for us?
Yeah, that's what's so valuable about mice, is that we've learned how to genetically engineer them. And they're one of the smallest mammals that we can do that with. At MIT, one can also do that with larger monkeys to study things that are more complicated, like social interactions. So the interactions of primates are really special, whereas mice don't have such complicated social interactions. And so depending on what you're studying, you might want to control different neuronal types to understand, for example, what goes wrong in human disorders. So now um, there are thousands and thousands of genetic mutations known in humans that disrupt everything from social interaction and autism, for which a monkey model might be appropriate, to Parkinson's-like symptoms that we can model in a mouse to ask what exactly went wrong, which neuron was disrupted. Can we repair that particular neuron and solve the whole problem without having to try to treat the whole organism with a drug? Those kinds of questions. And so I think there are several powerful things to mice, but one thing is just is being able to put these mutations that we discover in humans that have a known disorder into the animal to try to develop therapies and understand what's happened.
Right. And very quickly, with the genetic engineering, this is part of it. You can sort of manipulate this in the mice. Is that it? And get this reflection of the human thing.
Yep.
Right. This is very interesting research. And I understand that that this takes a very long time. I believe this is under the aegis of like basic research, right? And there's a real need for that. Does it take it a long time, years to develop these models and test things?
Yes, even making one mouse model can take as long as a year just to even make the mouse. Things have gotten a lot faster with CRISPR.
Yeah.
So that has been a revolution to engineering. And not only is it a revolution in terms of making it much faster to be able to make the changes, but it means that they can be made in any organism. And so we used to really be stuck with mice because people had figured out how to do it in mice. But with CRISPR, you can apply it to any organism. That's important because, as I said, mice aren't a good model for more complicated human disorders.
Right. That, of course, CRISPR gets rave reviews all the time. It has made a lot of work very much easier, much faster, and so on. So that's a good thing. I have one more thing before we leave. I know that you have been very much involved in a scientific community a project across a number of institutions to provide masks, to develop masks, provide masks for the COVID pandemic. Briefly, could you give us a little idea of what you're up to? Because scientists really need a pat on the head for, just dropping everything and trying to help out at this time. Please.
Thank you. Yeah. In March, I became quite active in this area, and MIT closed not quite long after that, as many other institutions did. And so scientists across the US found themselves not in the lab and wanting to help. And so people gathered together quite quickly on Zoom and started having, one group I was part of was having daily meetings to try to understand masks, how they work, and how to clean them, in particular to help the physicians who were short of these N95 respirators. And so the question was, how can you clean them? And so that was one of the first things I got organized in. And I think it's also very relevant to the public. And so that was another thing I did was try to educate the public. And I'm still working very hard on that because I think there are a lot of misunderstandings. And so I'd like to share that one thing I think that is really practical and important to understand is that it's when you're talking that the most particles come out of your mouth. And there's more that come out of your mouth when you're talking than when you're just opening and closing your mouth without talking. And more than when you're breathing through your nose or through your mouth. So it's particularly important to wear a mask while you're talking. And I think that's kind of counterintuitive to a lot of people. And also it means you need to have a mask that doesn't muffle what you're saying. And some of the best masks actually don't muffle what you're saying because they've been developed scientifically to have filters that can allow air to pass through them, but actively trap these respiratory droplets that might be carrying virus.
Very interesting.
And so those are examples. So sometimes you see the blue rectangular masks, those can
Yes.
be rated. So if you get ones that are rated ASTM 1 or higher, it's a rating that shows that they're trapping a lot of respiratory droplets, but not impeding airflow. And that makes them easy to talk through because they don't impede the airflow. And so people should have masks that are cover their nose and mouth and fit snugly. I think that's pretty well communicated. What I think is missing is that it's really when you're talking that it's most important. So even if you're dining with somebody, don't take your mask off as soon as you sit down at the table.
Right.
Just take it off when you're actually eating, and then try not to talk while you're eating, like your mom told you a long time ago.
Right, we'll have new motor habits. You'll have to do training sessions for that. It is a learning process, but this participation of the scientific community, drop everything and try to help out in this emergency period, is really awesome, and you deserve a lot of credit for that. Thank you very much. But also thank you for reminding us how important and why the masks are important at a time where we seen this like building resistance, developing resistance out there, and we need to all grow up and do our very best. It is frustrating, but Dr. Crittenden, thank you very much for joining us today and for all this very wonderful information. And I hope your research can resume again in a normal fashion in the coming year. Thank you.
Keep in good health. Thank you for having me.